he has developing for his PhD. He's supposed to finish in the next month. So he's presenting just one part of his work about bio-inspired feature descriptor. So that's it. So Steve, you can start your presentation.
Yeah, I'm going to share my screen. Please just confirm that it is being shared. Can you see my screen?
Yes.
Yeah, we can see it.
It is changing?
Yeah.
Yes.
Okay, all right. I'm honored with your presence today, and I really think I would like to extend my gratitude to Professor Christian and Eric for the opportunity given to me to present this noon a part of my PhD research about ecological diversity indices and biodiversity measures for texture characterization. So this is a part of the research that will be presented. Okay, so this is my description, the description of the work. What was the aim of this work, and how did we solve the problem? How we did carry out experiment evaluations and so forth. So the main aim of this work, research work, is the design and implementation of a new texture descriptor. And we have used the biodiversity concept and taxonomic indices, so which is a part of biology, in order to build a texture descriptor. And we have evaluated our work with natural images and histopathological images of real world and representative data sets. And we have used different quantitative measures such as accuracy, kappa coefficient, sensitivity, just to test the performance of our descriptor. And also we made some statistical analysis for verifying whether or not our descriptor performs well, not only in terms of accuracy, but also there is a statistical difference between this descriptor and other descriptors. So this is the agenda. So I'm going to introduce, to present some motivations and some assumptions that we have made, as well as the objective of the work. So I'm going also to present a bit of the theory behind this descriptor. So it is mainly the biological concept. And I will also present the proposal with some case study that we have applied to verify the performance of the descriptor. I will show some experiment results and some conclusions. Okay, so first of all, if in this work, our goal, we are talking about the texture. And if we try to define a texture, it is evident that there is no consensus with regard to the definition of texture. But a texture can be seen as a property of any surface. And it is variable information that can be associated with the interpretation of patterns related to various areas of object recognitions. And a texture also forms a non-deterministic complex system. For the texture is also referred to as a repetition of simple or complex patterns on a surface. So these patterns can be identical duplicates that appear repeatedly, or they can have some minor or non-deterministic change. So texture is a way to interpret visual informations using a natural characteristic of human eyes. So for a human eye, human eye has a facility to describe the texture, of course, under certain conditions such as illuminations, display scale, reflectance of the surface. So it is important in many applications to measure the texture in such a way that the values thereof can be transformed into useful informations, such as the identification of material, identification of the material of a surface, or in terms of in a medical imaging anomalies, for example, on microscopic tissues. So now we have, this is the perception of textures. So we have micro texture and macro texture, micro texture and macro textures. So we also have some subcategories of it. We have directional, we have non-directional, we have smooth, vice root, and so forth. So as we said that this, we can figure out, or we can observe or notice here that we have certain patterns, and these patterns can be, as we can see, identical duplicates in this case, for example, that appear repeatedly, or they can also have minor or non-deterministic change. For example, in this case, there are some changes that are non-deterministic, so we cannot predict how it will vary. Now, when we talk about texture analysis, here we seek to determine the neighborhood relationship of the texture element and their position in relation to the others, which is a connectivity. It also, we seek to determine the number of elements per spatial unit, which also we may call density, and the regularity, which also we may call homogeneity, therefore. And in the texture analysis, we have texture feature extraction and characterization, texture-based classification, we have segmentation, we have shape reconstructions. But in our research, we are relying on characterization and classification. So now, when it comes to the texture characterizations, it represents variable informations that can be associated with the interpretation of patterns to various areas of computer visions. But it is by and large difficult for most approaches to manipulate texture appearance while taking into consideration the effects, for example, of illumination, of viewpoint, and image surface shape, and also the influence of both microtexture and macrotexture to different existing type of textures may restrain the characterization process, as well as the development of method robust enough when both are found together or both are present in an image. So each of these type of texture, micro or macro, may contain intrinsic properties that may require a specific statistical methods to be represented to the maximum extent. So that is why sometimes one approach can work for a type of texture, macro, but not for micro. Now, feature extraction, when we talk about feature extractions for texture characterization, descriptors fall generally into two categories, which are statistical and structural. Now, when we talk about statistical, here we attempt to determine how some image properties related to its texture may be distributed, and then we can derive numerical texture measures from the computed distributions. So there are various attempts to characterize texture in the literature, the various attempts have been made in the literature over years, with the majority of them focusing on extracting the first order and the second order statistics. So when we talk about the first order statistics, so we mean the properties that can be derived directly from an individual pixel. So without any cross comparison between pixels. And the second order statistic is when properties involve comparing two pixels at the same time. And so this investigates how one pixel at some reference locations relates statistically to another pixels at a location displaced from the reference locations. What a bit tricky this. Now, in the structural, the aim of the structural is to investigate the different type of periodicity in an image and characterize a texture using the relative spectral energy at different periodicity. So this periodicity analysis is localized to the immediate neighborhood of the individual pixels. So, but we also have this. So we also have. Okay, now it's working. We also have model-based. We also have transform-based. We also have CNN-based method for texture characterizations. And here we have some example of each type of. Now, the motivation of this work was or is the development of techniques for texture analysis and characterizations in not only natural images, but also in those images containing other structure in addition to texture. For example, histopathological images, we have different structures, but we also have texture. And the adaptation of and development of strategies that will enable analysis with as much relevant information as possible. So we need to characterize texture to the maximum extent. And the characterization of image texture, we want to characterize a texture from an image with some essential corporates. So we want to develop a texture descriptor that will be invariant to change in image contrast, invariant to in plane rotation of the image, and descriptor that will lend itself to fast computation. So, and also how we can apply machine learning technique to the processing of patterns in a generic manner. So now here are some objectives after these motivations. So the overarching objective of this work was or is to present the development of approaches for extracting features from a texture image that are generic, that provide a high accuracy rate, that are independent of micro-level variation in terms of contrast, that are invariant to in-plane rotation of the image, and lend themselves to fast computation and can be explained and interpreted. So we have three sub-objectives, which are to investigate, adapt, and apply biological concepts, which are taxonomic indices and biodiversity measurements for characterizing texture from an image, and to propose new texture descriptors based on biological diversity measurement. And also, we would like to compare the performance of the BIT of the descriptor that we are proposing against classical method and some deep ones in the literature. Now, before presenting our assumptions, so our point of view is firstly based on the principle that the principle that data distribution of textures forms a non-deterministic complex system, which means that its behavior cannot or not always be predicted. So we state that the textural patterns behave similarly to ecological patterns. In ecological patterns, large population of units can self-organize into aggregation that generate patterns from processes that are non-linear and non-deterministic. So therefore, our study investigates the following hypotheses: the use of information theoretical measures of ecological diversity indices together with measure of biodiversity can provide a robust tool for quantifying such a complex system of diverse patterns that we find in the image, this which means the texture. And such a tool, which means the descriptor that we are proposing, will have both statistical and structural approaches, properties or capabilities, and will take advantage of ecological patterns invariance, because the ecological patterns are invariant to permutation, to rotation, and scale by employing the concept of species richness, abundance, evenness, and taxonomic indices. So these are our hypotheses. Now we are going to go to have an overview of the biological concept that we have applied in this work. First, let's define what is a biodiversity. Biodiversity is defined as, this is one of the definitions, there are many different ones. So the biodiversity is seen as a variety within and among life forms on a site, ecosystem, on a landscape, or on a unit. And several attributes can be measured to describe ecological communities, which include density, frequency, cover, and biomass. And there are several ways that these individual attributes can be combined to create descriptive measures of ecological communities. So this includes, if we combine density, frequency, cover, biomass, so we may get a dominance, diversity, composition, similarity. I may be using the term biology or biological and ecological interchangeably, but biology is, as we know, that is the study of the anatomy, physiology. Ecology is part of cellular and molecular biology of all living things, whereas the ecology focuses on the environment, climate, and natural resources and how animals and plants respond to these factors. So if I use the term ecological or biological, so it's not the same, but it's in the same line. Now, why biodiversity is measured? So this is a big question. So the biology is often measured because it is believed that high biodiversity is an evidence of ecosystem health. So generally, diverse communities are said to have are said to be stable, to have an increased productivity and resistance to invasion and other disturbances. Now, in biology, biodiversity indices are often used to estimate the consistency and distribution of a species community. Now, the idea of such indices is to obtain not only the quantitative, but also the qualitative estimate of biological diversity or variability in space or in time that can be afterward used to compare biological entities composed of diverse components. Now, only a multidimensional assessment of the biodiversity can provide an insight into the relationship between change in biodiversity, change in ecosystem functioning, and change in ecosystem services. It is said that there is no feature on Earth more complex, dynamic, and varied than the layer of living organism. Now, another question is, where is biodiversity? Where is biodiversity found? Okay, where is biodiversity found? Where can we find the biodiversity? So, the biodiversity is found in ecosystem and ecoregions. We have what we call spatial patterns of biodiversity, where many patterns and tools which make patterns and tools that decision makers can use to derive useful approximation for both terrestrial and marine ecosystems. And we also have temporal patterns of biodiversity, which is knowledge of which consists of the knowledge of patterns of biodiversity over time that allow for background rates of extinction and biodiversity lost. So now we have here three types of indices that we can find in biodiversity. So we have indices that we call species richness indices, so which is a measure for which measures the total number of species in a site. We have evenness indices, which express how evenly individuals in a site are distributed among different species. We have taxonomic indices, which take into account the taxonomic relation between different species or organisms in a site or in a community. So the taxonomic indices or the taxonomic diversity reflects the average taxonomic distances between any two organisms that are chosen generally in random from a sample. And the distance, when we talk about distances here, the distance can be seen as the length of the path connecting these two organisms along with the branch of phylogenetic tree. So I'm going afterward, later on, present an example of a phylogenetic tree to better understand this part. So now, there is a difference between richness and evenness indices and taxonomic indices. First of all, sometimes we may think that sometimes when we talk about species richness, there is a confusion. We may believe that species richness is the same as diversity, but it isn't, because diversity implies or involves both richness and evenness, which makes the diversity. Now, the richness and evenness are based on abundance, whereas taxonomic indices are based on the parenthood relationship, distance, and the distance there here are based on phylogenetic diversity indices. These days, I have been thinking, after reading much content from literature, that the taxonomic indices may be what we call, may be a third order property. because if it is so, then it will involve, for example, investigating in the image context the gray scale and color distribution at 3 or more pixels. I will talk about later, but I've been thinking of it and sharing some ideas with some experts in biology and in ecology, but it is not clear yet to me whether it can be considered as a third order statistic. But it's tend to be after the experiments that we have carried out. So we have this perception of this taxonomic to be also the third order statistic. Now we also have three different spatial scales that the 3 indices presented follow. We have alpha diversity, we have beta diversity, and we have gamma diversity. So the alpha diversity refers, the alpha diversity refers to the diversity within a particular area, community, or ecosystem, and usually in a species level. And we have a beta diversity, which is a diversity between ecosystems, and we have gamma diversity, which is a measure of the overall diversity for different ecosystem within a region. Now we have here different biodiversity indices that we have used. Here are some examples of the first part of our research, but we have other 9 or 10 that are not presented here. So the diversity indices that we have used can be cataloged into three classes. We have homogeneity indices, we have information theory indices, and we have equitability indices. So some example we have here are, we have Markolev's diversity index, Moninick diversity index, Berger-Parker, Fisher alpha, and so forth. And we have also McIntosh evenness, we have Shannon Winner, both are information theory. Both are information theory indices. And we also have taxonomic indices that we have taxonomic diversity, we have taxonomic distinctiveness, we have some of phylogenetic distances, but all these distances are based on, for example, this one are based on the species richness, abundance, and evenness, and those are based on the distance. So we are going to show how to apply all this concept in the image level. So here we have some definitions or some translations how we have represented our image as an ecosystem. Because if we want to apply the biological concepts for texture analysis, there should be a one-to-one correspondence between image analysis terms and ecosystem terms. So whenever we have ecosystem sites or ecosystem site units, it is seen as a whole image or part of it or a region of interest. Species are considered here as the number of gray levels in the image or a region of interest. Individuals are considered pixels of an image or region of interest, and species abundance, number of pixels of a particular gray level in the region, and species richness is the number of different gray levels found in an image or a region of it. So here we have an example. So in this example, we have an example of a site or 3 sites or 3 ecosystems. let me say so. So we have the site A, site B, and site C. So the site A, we can see, so if we count the species, we have we have seven species. So this element are species. We have site B, and the site B has 5 species, and we have a site C has 7 species. Now here, these are alpha diversity, okay, of A equals 7, alpha diversity of B equals 5, alpha diversity of C equals 7. And what about the beta diversity? So if we observe the beta diversity between site A and C with 10 species, which is the total number of species we have, that differ between them only 2 species in common. So if we see B and C, they only have 2 species in common. And if we want to have the overall idea, which we call gamma diversity in these 3 communities, so the total number of species is 12 species total. So the diversity, and so we have 3 sites or 3 communities, and we have just 12 in total number of species. So now here we have an image example. How an image can be represented as an ecosystem is, let's take this gray image as an abstract model of an ecosystem. So here we can see that we have 3 gray levels. We have white with 6 individuals. We have gray with 5 individuals, and we have black with 5 individuals. Now from this, we can now see, first of all, from here, only with the abundance, we can compute the biodiversity measurement. Now, to compute the taxonomic measurement here, we need exactly this. So we present first an example of a phylogenetic tree, which is this one. So we have 4 species A, B, and C, D. So in this phylogenetic tree, we can derive a distance matrix. For example, we have A and D. So we can count 1, 2, 3, 4, 5. So the distance with C we have 1, 2, 3, 4. So the distance is 4 here, and so forth. So if we can do that, it means how can we apply this concept of having a phylogenetic tree to construct the distance matrix from an image? From an image, what we did was we have an image here. So with the values of, we have our image, and we have gray levels and the corresponding values. So what we did is we divide, we are going to split this image in order to obtain the leaf. So here we have leaf. So we want to have these leaves. How can we have them? So we need to split our image using the mean value. So if we compute the mean value as our threshold, so all the value below will go to the left, and all the values, all the gray levels, all the pixels above this threshold will go to the right. So we make these divisions recursively until we get a species that are called leaves, and we can now build what we call the phylogenetic tree, or the dendrogram of the corresponding dendrogram of this phylogenetic tree here. So, for example, which means that here we have the species 6 and the species 75. So we can compute their distance. We have six individuals of the species 6. We have 6 individuals of the species 75, and the parenthood of both species is that both species are related. And what is the distance between them? The distance is 2. It's just 1 and 2. The distance between both of them is 2. So here we can build in our matrix. We have 6 and 75. So the distance is 2. And what is the distance between 6 and 230? So distance between 6 and 230 is 5. How can we compute this? We have 1, 2, 3, 4, 5. So this is how we are counting the branch that are separating, or the branch separating both species in order to establish the value that we are going to use in our distance matrix. So we cannot apply taxonomic indices without firstly mapping this image or transforming an image to a phylogenetic tree or dendrogram. Afterward, construct the distance matrix from which we can now compute our taxonomic indices. So now, what are the properties of the descriptor that we are constructing based on this measurement? First of all, I said in the very beginning that a textual descriptor should have some essential properties such as invariance to rotation, invariance to scale, invariance to intensity change, invariance at intensity change, at least a monotonic intensity transformation for different applications in computer vision. And also the descriptor should be easy to compute. Now, the diversity indices that we have shown here based on richness properties directly relate to species. They are naturally in biology, they are invariant to in-plane rotation, invariant to scale, and also they are diversity is invariant also to the species abundance vector permutations, which makes and also the BIT descriptor combines statistical and structural from our, because the structural from our statistical from the from both taxonomic indices and biodiversity measurement. And the BIT, so the descriptor that we are proposing, we name it BIT, bio-inspired textual descriptor. This descriptor takes advantage of ecological patterns, as I said, invariance to permutation, to rotation, and scale by combining richness, abundance, evenness, and taxonomic indices. Now, we are going to present here a table. So what is this? We carried out some experiment just to show how we have an original image here, and after some transformation of the image, we compute the value of each indices in different transformations. So we have rotations, we have reflection, and we have rescaling. So if we observe here in this table, we can see that the value, for example, of Margaret is the same for regardless of the transformations, but it's not the same, for example, for rescaling. So for rescaling, it fails. And we have others here. We purposely put the indices that fails to maintain invariance in scale. We have one here that maintain the invariance in scale, but we have others, 9 others indices that we have used in another part of the work that are even invariant to scaling. So here we took the example of 2 images. We have a natural image, and we also present histopathological images. So just to show the invariance properties that the BIT inherit. Now, the BIT also shares some common characteristic of other textual descriptors, such as we have a GLCM and the LBP, because the BIT characterized texture also based on second-order statistical properties. And in ecology, taxonomic indices are approximations of a second-order statistic at the species level. And the BIT also shares some characteristic with Gable filters. As we know, Gable filters explore different explore different periodicity in the image and attempts to characterize a texture at a different periodicity. In the same line, the phylogenetic tree also combined with the diversity indices are used in biology to compare behavioral patterns between species in different areas and within a neighborhood. So there is also something important to say is that the diversity indices based on species richness are very important when defining the all-inclusive behavior of an ecosystem, because they do it well, taking into consideration that the ecosystem's patterns form a non-deterministic complex system. So now we are going to present a case study that we have used in this work. So we compared the performance of our BIT descriptor with GLCM, Haralick, and LBP, and also with some state-of-the-art, with some works found in the literature. So this is the scheme that we have followed. So when we have an image, we split an image into different channels because we also want to explore the color information. And after splitting, the image may undergo pre-processing, and after we extract features. So since we are comparing the GLCM, Haralick, and LBP, so we use the same process. For the BIT, we extract BIT, and we use the same pipeline to extract GLCM, LBP, and so forth. And for each descriptors, for features from each channel, for all the channels are after combined, just to make a single feature vector. And then we normalize, but the normalization was different. So what we did was, first of all, we split. Okay, so we split the dataset into training and testing. We normalize the training, and we use the parameters of training normalization to afterward normalize the testing. So we did not normalize all and split. So we just want to reflect the reality, because in the reality, the testing are not necessary. The testing may be image may be captured, for example, from with a different camera, let's say. So we just want to reflect the reality. That's why we make this. We use this process. So, and here we have the images that we have used. So we used this texture images. We used. We have. Can you see the images here?
Yes.
Okay. So, okay. We have here Salzburg dataset. We have Altex dataset. We have KTH dataset, and we have histopathological images. We have Brekis, and we have CRC with H class, and we have here natural images. Now we are going to show. I'm going to present quickly because I remember that I do not have 2 hours. So I have to go quick. So here we present the average accuracy of the average accuracy of different approaches, descriptors that we have used. Here we have the accuracy of for Salzburg. We can see that we have obtained the best result of BIT with superlearner, of Haralick with SVM, of GLCM with KNN, and of LBP with superlearner also. And we have for Altex, we can see that for Altex we have 99 99.88% of accuracy with the BIT. With Haralick, we have 96, we have 94 with GLCM, and we have 83 with LBP. And for KTH, we have 97, 97, sorry, 97. We have 94 for Haralick. We have 76, and we have 60. We have 64 for LBP. But also, what we wanted to show is that BIT also performs well regardless of the intensity variation. So we have applied the gamma correction to the testing, okay, to the test set. So applying gamma correction to test set. So here we only compare. Oops. So it is giving this. Okay, I don't know how we do this. Okay, so here we compare the best result only obtained, okay, with for each data set with the gamma correction applied to the test. So we have applied the gamma correction of 0.5, 1.5, 2.5, and so forth. So this is the result that we have obtained. We can notice, we can note that for Salzburg, we have lost again. Yes, there is yes, there is I can say drastic change. For example, with gamma 3.0, we have lost at least 4, okay, nearly 4%. But for Altex and KTH, so we can see that we have the result decreased, but slightly decreased. So we can say that BIT also performed well. So regardless of the intensity, the illumination intensity. So here we compare the BIT with some related works in CRC using 8 classes. We found many classes, many works with 4 classes, with 2 classes, but because we are using 8 classes, we compare with this work. So we can see that we got for 10 folds, we had 92%. So here 92%. And for 5 fold cross validation, 92 also. And we have another work here with 97. This work used 2 classes, and our intention was to use 8 classes. And we also compare the result with the BIT. So with the BIT, with the breakist data set, the BIT with SVM presented the accuracy of 97.29 for 14 magnifications, because this data set has 4 magnifications, but it lost for 100 magnification, 200 magnification, 400 magnification against the work using the work that used CNN for the 3 magnifications, and with a slight or considerable difference. It depends on the interpretation. But I personally was satisfied because, as you know, that CNN is almost divine, even though for texture, CNN in the literature did not perform quite well for texture analysis. But the difference also is that the BIT here has a low computation. We do not need a bunch of parameters as the CNN requires, even though there are some CNNs architecture that requires less parameters, but it takes time and also needs a bunch of data that in the context of medical images. So we have it is a bit restrained to have a bunch of images. And the reason also to test the BIT in different type of data sets, medical and natural, is just to prove that BIT is a generic one. So for summarizing, so we have revisited several biodiversity measures and taxonomic indices to compute feature-based, to compute or to form a feature descriptor based on the richness of species, the abundance of species, the evenness of species, and taxonomic indices. So we combine all these components to and to take advantage of the invariance characteristic of ecological patterns. And this feature descriptor form a robust and invariant texture descriptor that can be used together with different classifiers to build a powerful classification model. And it has a low computational cost and compared to some state-of-the-art works, and especially with CNN. So it is promising. It achieved a very competitive result compared to deep method, even though currently we are working. So we have been working since the first version of this work, of this, I can say even this paper. We have been working hardly, and we came up with about 10 new indices that outperformed even the result that we had in the first version. So Bing, I thank you very much. I believe I took much time, so sorry for that. So this is what I wanted to present about the research we have been conducting. So thank you very much for your attention.
Yeah, thank you, Steve, for the great presentation. So it's a little bit late. We're running out of time, so maybe we have time for one or two quick questions.
Hey there, Matthew Tates here. I liked your presentation. I'm just wondering, did you see that DTD data set that I put in the chat there? There's a whole bunch of like CNN papers that have been using this one. It's quite popular, and I'm wondering if you could maybe evaluate your system against this type of data and approaches. I'm wondering if you looked at this one.
Okay, I would just say, okay, because I couldn't see the chat when I was presenting.
Yeah, no, I just put it. Put it. Yeah,
okay.
Yeah. It's just from Oxford University in England.
Okay. Okay. Thank you very much. I will take it. We have the. Okay. Thank you for the suggestion. So we will.
Thanks for the presentation.
Okay. Thank you, sir.
Eric, you have a question?
Yeah, well, thanks, Steve. That was really interesting talk. I enjoyed kind of the process you had, the bio-inspired and all that. I'm just wondering, like, so you're using a descriptor, and there's no learning involved in the production of the descriptor like LBP. It's just there's an image coming in, you produce a representation. So it's like a computer vision type approach. And I'm just wondering how it compares to to learning approaches, because I mean, I guess in recent years, people have gone heavily into, deep learning or learning representations with bigger models. And have you done any comparisons, for example, if you used a convolutional network to produce representations, or?
No, I didn't.
Okay.
But I guess it's a good, thank you. I believe it to be a good suggestion.
Yeah, I would assume it, depending on where you publish, you would meet that kind of question, I think.
Yeah. So here we used we use the BIT, so these indices to do the same thing as the GLCM does, for example, as Haralick does, where it has a compute the homogeneity, the contrast, and so forth. So we did exactly the same thing, but in the biological concept, where each result we can give an interpretation based on the biological concept.
Right.
But
right.
I believe that this of comparing with representation learning with CNN yes, he's a good go.
Okay.
Thank you very much.
But I've often wondered, like, I think a lot of people, I mean, this is somehow a bias. Often people who use, for example, who, you know, have some learning approach to create a representation, even if it's something like PCA, they use to reduce features or improve their features. It's somehow you're using data. And in a lot of experimental methodologies, people use data, their training data to, and even sometimes more, you know, even the validation data to create some sort of a translation matrix from the original space to the representation space. So they're already using data to do that. And then they're using it again to do training of their model, their classifier, let's say, or other models. So somehow there's a bias. So, I mean, I think there's a place for descriptors in that sense. And I find that in some of the works, there is that bias when people use all their training data to create a representation and then they train a classifier, then they, you know, so I think, anyways, I'm generally, I think in your case, that bias doesn't exist, but just another comment, if we have time, or if there are no other questions, I'll ask Christian if there's any other people.
I don't see anyone else, so maybe we can,
Okay.
can maybe finish with that question.
So one of the questions I had as well is, so you're interested in diversity, and so you're using a measure of biodiversity that is based on images. So that's kind of an interesting approach. I'm just wondering if you've considered this for learning classifier ensembles, because like when you learn a classifier ensemble, I guess there's a few steps there. The first step is you want to generate a pool of classifiers that is diversified. Then you want to whatever, maybe select a subset and learn a fusion function, or you know, that sort of thing. But in the first step, you're supposed to, when you're training an ensemble or learning an ensemble, you're trying to generate diverse classifiers. And I'm just wondering if these diversity measures that you were presenting would be a good way to select those classifiers or to create them, you know? Do you have any thoughts on that, or?
Yeah, to create them, I was discussing with my supervisor. We were talking, and we came up with this idea that what if from using the biodiversity indices, we can construct, okay, we can help to improve a classifier. So that, I guess that we were discussing, because we have 3 levels. So, look, we have the alpha diversity, we have the beta diversity that we compare to ecosystem, and we have the gamma diversity that will have the overall idea. So from this concept, I believe that it will be, I assume it will go wrong, but I believe that it is something that deserves an attempt, because that deserves an attempt.
Yeah, it sounds promising.
Yes.
Seriously, yeah.
Good. All right, well, let's thank Steve for the great presentation.
Oh, thank you, sir.
Thank
Thanks
you.
again, and
Thanks, Steve.
yeah, good luck for your, I understand that you have your thesis defense coming up soon, so good luck with that.
Oh, thank you very much. And
yeah, hope to see each other at the lab. That goes for everyone. I mean, it's always nice to have to meet face to face at the labs. So
I'm looking forward.
All right, so have a great afternoon, everyone, and I'll see you probably in 2 weeks for the next seminar.
Okay, thank you.
Bye.
Bye now.
Bye.
Bye.
Bye.